So now let's look at this hormonal control that I've mentioned quite a few times. So looking at this image, on the left side, you see food traveling in. It goes to your stomach. Then it becomes chyme when it's dumped into your duodenum. And then as it passes through your duodenum, it arrives at the jejunum. So what happens when the food is in the stomach? Well, gastrin is released, and that allows for acid production and gastric motility, which is the movement of the stomach. Once the chyme reaches the duodenum, we have GIP, which is gastric inhibitory hormone, secretin and CCK are released, and VIP is released. And so each one of these hormones has a different job. The GIP, insulin release. Secretin and CCK, enzyme and buffers, and then bile secretion. VIP, dilation of the intestinal capillaries. In order to be able to accept anything, the capillaries need to be dilated so that substances can enter. When that dilation occurs, it facilitates nutrient absorption at the level of the jejunum. Going back up to GIP, once you get the insulin from the pancreas, it again facilitates nutrient absorption. All right, so this image gives you a very brief introduction to what the hormones do. But I think it's incredibly helpful for you to have an overview of these 5 hormones and what their overall job is. All right, so these are peptide hormones. They have different effects, as you saw from the previous image, and it depends on what region of the digestive tract you're in, what the effect is going to be. Secretin is released when chyme reaches the duodenum. And when this happens, it allows for an increase in bile and buffers. Cholecystokinin is secreted in the duodenum. So this occurs when you have lipids and partially digested proteins. It allows for the production, accelerated production of some digestive enzymes. Also, CCK has the job of relaxing that hepatopancreatic sphincter and the gallbladder. And so this allows for bile and pancreatic juices to make their way into the duodenum. The GIP, the gastric inhibitory peptide, occurs when you have fats and carbohydrates enter. And the vasoactive intestinal peptide, vaso, of course, meaning vessel, it dilates these capillaries in specific regions, and it also inhibits the production of acid in the stomach. Gastrin, we go back to where we were looking at plica and how when we were looking at the different cells in the small intestine, it's secreted by G cells in the duodenum when you have incomplete or partially digested proteins. It increases motility, so it allows for movement, and it stimulates acid and enzyme production. So when you ingest that peanut butter and jelly sandwich, it's going to take about 5 hours for the materials to go from your duodenum to the end of the ileum. So it takes about 5 hours for that movement in your small intestine. In order for this movement to occur successfully and for you to get all of the nutrients that you should have, you have to have this movement to stir and mix the contents. And not only does it stir and mix the contents, but it changes the environment around the epithelial cells. And something that I would like for you to remember throughout all of what we do, remember that when we're talking about digestion, remember that we have central nervous system control, we have endocrine control, and remember that we also have something called local control. So what's going on locally is incredibly important. Right, now moving on to the large intestine. It is large, it's a large horseshoe-shaped structure. It's about 1.5 meters in length. It goes from the end of the ileum, as you can see right here, the end of the ileum. It frames the small intestine. Remember, the small intestine has been removed from this image so that we can actually see the large intestine in detail. And sometimes it's called the large bowel. The job of the large intestine, or the jobs, reabsorption of water. And as you know, if you have had a bout of diarrhea, water has not been reabsorbed effectively. Also, the compaction of the intestinal contents, removal of or absorption of vitamins, and the storage of fecal material. There are 3 parts. We have the cecum, which is this area down here, the colon, which is the largest area, which you can see right here, and then we have the rectum that you can see right here. It's the last portion of the tract. So this picture down here at the very bottom, I think is great for you to get reference size. I want you to look at this person's thumb and then look at the size of the appendix. They are about the same size. Also notice this structure right here, which is called the ileocecal valve. Do this again, the ileocecal valve. And this is the cecum that you can see right here that has been cut open. So the ileum, the last portion of the small intestine, attaches to the cecum, and it opens up at the ileocecal valve that I just pointed at you. So the cecum itself is part of the large intestine. It is an expanded pouch. It receives the material from the ileum, and it starts the compaction and stores material. The appendix that I showed to you is about 9 centimeters long. Sometimes it's called the vermiform appendix. So if I asked you what the language of science was, you might say Latin, you might say Greek. The modern, and I'm using air quotes for modern, language of science was actually German. So giving you that hint, if you see the word vermiform, what does that mean? Well, it actually means worm-like. Verma is worm. It has lymphoid nodules, so it's considered a lymphoid organ. The use of it is not really well understood. It could be a vestigial organ, and you know that it connects in the cecum. The colon itself, it has a larger diameter but thinner walls than the small intestine. The walls of the colon form these pouches. So these pouches are indicated, let me change color again, indicated here. Each one of these is called a hostra. These are the little pouches, and they permit expansion and elongation of the colon. There are 3 longitudinal bands of muscle, so you can see them right here. These are called the taenia coli, and that allows for the colon to contract and to expand as necessary. Because this is a muscle, like any other muscle, it needs to have tone. And someone who has not been on a solid diet for a while, maybe they were in a coma or they had to have some type of surgery, can lose tone in their colon. And so it takes time for that muscle tone to come back. So if a person is taking opioids, opioids slow down all processes. And so it makes it very difficult for a person who has been on opioids to maintain the tone in their colon, because oftentimes they lose the tone, and constipation is a significant issue for them. So there's a serous membrane of the colon. It has these drops of fat. Here is the cutaway of the serous membrane, and here are the drops of fat. They're called epiploic appendages. There are 4 regions to the colon. We have the ascending colon, the transverse colon, the descending colon, and then we get to the sigmoid colon. The rectum is the last 15 centimeters. It's expandable, and it allows for the removal of fecal material. The anal canal is the last portion of the rectum, and it has longitudinal folds that are called anal columns that disappear when filled and will reappear when empty. The anus is also called the anal orifice, and it is the exit of the anal canal. There are 2 sphincters in the anus. The internal anal sphincter is smooth muscle, and it is not under voluntary control. The external anal sphincter is skeletal, and it is under voluntary control. So when you think about potty training a child or potty training a dog, they're not under control of that internal anal sphincter, and it takes time for neural networks to be developed for them to gain control of the external anal sphincter. That's why you can't potty train a dog at 4 weeks old or a human at 4 months old. It just takes time for those neural pathways to be developed. So the colon lacks villi, so that tells you right there that it has a different job than the small intestine. The glands are deeper than the glands of the small intestine, and there are many, many goblet cells there, so there's a tremendous production of mucus. The large intestine does not produce enzymes, but it does produce lubrication for fecal feces. In the large intestine, there is the reabsorption of water, the reabsorption of bile salts that then go back to the liver. You have absorption of vitamins, and organic waste are absorbed. In the large intestine, you do produce vitamins, vitamin K, which is fat soluble, which is necessary for clotting. Biotin is water soluble and is necessary for glucose metabolism, and then pantothenic acid that helps with the production of neurotransmitters and steroid hormones. You know that organic waste are managed in your large intestine, and in your large intestine, your good bacteria are going to convert bilirubin, which are urobilinogens, into stercobilinogens. Feces that have peptides in them are going to be broken down by, or the peptides are going to be broken down by bacteria, and that produces ammonium. Indole and skatole are actually the compounds that are responsible for the odor of feces. And you also have hydrogen sulfide that is going to be produced by this breakdown of peptides by bacteria. So because of our good bacteria, we get ammonia and indole and skatole and hydrogen sulfide. The bacteria in your large intestine like those indigestible carbohydrates, those complex polysaccharides, such as beans. And that is what allows for the production of flatus or intestinal gas. So whenever someone has intestinal gas, it is said that they are flatulent, and it comes from the clinical description of flatus. All right, so now I have popped up this image, and I don't want you to just look at it and go, oh my gosh, I don't know anything about it. I want to reassure you that you actually do. Because when we learned about large organic compounds, such as carbohydrates, lipids, and proteins, you learned about what they were broken down into. So let's just pull up that information and think about where it happens. So here on the left side, you can see the different areas of your digestive system: oral cavity, esophagus, stomach, small intestine, then the mucosa of your intestine, and then your bloodstream. All right. So what is broken down? What is digested in your oral cavity? Well, there's only two things, and it's the beginning of digestion. For carbohydrates and for lipids. That begins in the mouth. And you can see the enzymes that are used: salivary amylase and lingual lipase. Well, let's just take carbohydrates. So notice that nothing happens in the esophagus. No digestion is going to occur. But those carbohydrates that were beginning to be broken down in the oral cavity will then continue to be broken down once they get to the stomach. And notice that it is gastrin from G cells that allow for this breakdown to occur. Also notice that, and this is in the stomach, when we get to the small intestine, these disaccharides and these trisaccharides are going to continue to be broken down because we get pancreatic alpha amylase, right? And that is triggered by the hormone CCK. So what is the stimulus? The arrival of chyme in the duodenum. And what do you get? You get all of these enzymes that are produced. Please do not think that you need to know these enzymes. You do not need to know these enzymes, right? Those are proenzymes. But what I would like for you to know are these enzymes: pancreatic amylase, pancreatic lipase, those two for sure. Do not worry about nuclease and enterokinase, all right? I just marked it up for you. Then when we get to the mucosa of the intestine, we've broken down the disaccharides and the trisaccharides into lactase and maltase and sucrase. Then what happens to them? Via facilitated diffusion and co-transport, the monosaccharides that are produced are going to go into the bloodstream as monosaccharides. All right? So that's pretty simple for carbohydrates. Because remember, you learned this before, what happens to disaccharides and trisaccharides? They're broken down into monosaccharides, and they go into your bloodstream. So now let's look over here at the lipids. So nothing happens in your stomach with lipids in regard to chemical events in digestion. But once you get to the small intestine, bile and pancreatic lipase are going to break down these lipids into fatty acids. We didn't talk about micellus, but just know that the fatty acids are going to be diffused into the mucosa. We'll get another color. Diffused into the mucosa, and they're going to be further broken down, and they are going to enter the bloodstream. And proteins are pretty simple. Nothing happens in the mouth. Nothing happens in the esophagus. Polypeptides are going to be formed because pepsin has been produced. Once we get to the small intestine, you have these substances that are then going to break down the polypeptides into short amino acids, or sorry, short peptides and amino acids, which are then going to be further broken down and transported, but they're going to be broken down into their smallest component, amino acids, which are then going to enter the bloodstream. So just remember to take this apart, take it in small bites. You actually do know this information. So your digestive system breaks down the physical structure of food, either mechanically and chemically. It disassembles the component molecules and makes them into the smallest components that they can. Those molecules that actually go into the bloodstream are going to be absorbed by cells. And the whole purpose is to provide energy for ATP synthesis, right? And then we're going to synthesize more carbohydrates and proteins and lipids because we have this ATP available. Your digestive enzymes are going to be secreted by salivary glands in your tongue and your stomach and your pancreas. And molecular bonds are broken in the large organic molecules. And I just showed you that in the picture. And the process is called hydrolysis. So these hormones are broken down into their, these enzymes are broken down into their specific groups by the organic molecule target. Carbohydrates break down simple sugars. Proteases break amino acids. Lipases break down fatty acids. If you need some animation to help you understand the material, you can go to the Digestion overview on YouTube.